Thank you for a wonderful talk. Given that lunchtime is 12:30, you can think of our next speaker as the appetizer. So Dr. Amitabh Chak, he's the last speaker. He is a professor of medicine, Case Western Reserve University, and director of Advanced Technology and Innovation Center of Excellence, as well as director of clinical research in their GI division. His talk will be risk factors for esophageal adenocarcinoma, especially talking about those familial issues. Amitabh.
Thanks, Hashim. I want to thank the board members for allowing me to come and attend and present some of what we're doing and do it rapidly. I do have to disclose that I have some patents and biomarkers in Barrett's detection, including that booth that was in the exhibits, but we're not going to be talking about detection. What I am going to talk about is briefly cover the epidemiology of Barrett's and cancer, which they wanted me to cover, but then I decided I would spend more time on going through something that's near and dear to my heart, which is our familial and genetic studies. The risk factor for Barrett's and adenocarcinoma, most of you are fairly familiar with. I'll just try to summarize them quickly. There's some that we're born with, such as aging, male gender, white race, and family history, which I'll come back to. And then some that we acquire as we get older: reflux, obesity, smoking, H. pylori or absence thereof, and medications, which Stu discussed so elegantly in his talk earlier, and I won't discuss. The age incidence of Barrett's, of course, we can never know because you only find Barrett's when you diagnose it, but the prevalence was described by Cameron in this elegant study on a population in Olmsted County, and the age starts to plateau at about age 50 and leads to our guidelines for when we begin screening. And that's convenient because it's about 12 to 13 years earlier than the peak incidence of esophageal cancer, which seems to happen at about mid-60s, early 60s. The female ratio in this disease also is interesting. It tells us something about this disease. It's clearly a male-predominant disease, but if you notice as you dig out, the men-to-women ratio for Barrett's is somewhere about 3 to 1, but the men-to-women ratio for esophageal cancer is about 5 to 1 or 6 to 1, telling us that the male gender not only predisposes to initiation of Barrett's, but there's something about being a man that also drives that progression from Barrett's to cancer, factors that we do not know, and I'll come back to that. Barrett's is also much more common. It's primarily a disease of whites. It's seen more often in non-Hispanic whites. It's, of course, seen and reported in Asia, but much, much less a disease. And again, it tells us something about what's inherent about this disease. And of course, the symptom that brings most patients to our attention is reflux, but as shown elegantly in studies that David Lieberman did and many others in the past, it's not really the frequency of reflux or the severity of reflux that's important. Reflux can be relatively minor, but it's more the chronicity of the reflux or the duration of reflux, with the odds going up the longer you have reflux. 5 years is the cutoff that we use for our screening guidelines, and the more recent ACG screening guidelines take into account all those factors: male gender, older age of 50, and then duration of reflux of 5 years or more. Reflux, of course, is a risk factor for adenocarcinoma, and it does lead to our screening guidelines. And in this population-based study from Jesper Lagergren, it shows us two things. The odds for reflux in esophageal cancer is about 8, really emphasizing why we care about reflux. But the other part of this study, and the worrisome part, is that 40% of those with esophageal cancer had no prior history of reflux. So all the strategies that we've been talking about and even our screening guidelines are still not going to address those 40%, and why we have to think of other ways of identifying those subjects. Obesity was a risk factor that we had not thought about in the early days of Barrett's esophagus because it was so collinear with reflux, and it was heartburn that really brought people to our attention. But the obesity, Barrett's, and reflux trio had to be teased out by some elegant studies, these case control studies published in the same issue of Gastro, first by Doug Corley in the Kaiser population, where he found an odds ratio of 2 for those who had a waist circumference greater than 80 versus waist less than 80. It was really central obesity, something about that metabolic activity that led to Barrett's. And then in the same issue of the journal, Tom Vaughn did a case control study on the Seattle population where obesity per se, BMI, was not associated with Barrett's. It was only when you teased it out and looked at the waist-hip ratio that he found a high waist-to-hip ratio was associated with Barrett's. So it's something about the fact that obesity is truncal and metabolically active. And remember that in men, obesity tends to be much more truncal than it is in women. And maybe that explains why Barrett's is initiated in those individuals, or partly explains it. And then, of course, obesity per se, BMI, is a risk factor for esophageal adenocarcinoma, so that association is a little bit different. Central obesity with Barrett's, but general obesity with adenocarcinoma. Lots of studies that show an odds ratio of about 2 for association of obesity and cancer. And finally, the other important risk factors that we use in our screening guidelines is cigarette smoking. And in this meta-analysis of a number of case control studies, Michael Cook showed that cigarette smoking has a dose response, plateaus at about 30-pack years, and again, the odds ratio is 2, not as high as squamous cell cancer, but it is a risk factor for Barrett's also. Now I'll switch from those things that most of you probably also know, but delve a little more in detail into our family and genetic studies and where they are and where they're headed to. So let's think about it. You know, why postulate a genetic susceptibility? Barrett's was always thought to be an acquired disease, so why should we think something that incidence is going up? Why would you think there might be something genetic in there? Well, the epidemiology does give us a clue. This predominance of male gender, and especially the predominance of a specific race, are features that are often seen in other genetic diseases, and that's telling us something. We've talked yesterday a lot about reflux. Most of those patients with reflux will never develop Barrett's, and certainly very few of them will develop adenocarcinoma. So Barrett's appears to be restricted to a subset of those patients. Maybe there is an inherent susceptibility to developing Barrett's esophagus. And then in general, as our knowledge has advanced, as we solve the human genome, we've begun to realize cancers are not a single factor disease. They're complex diseases that include genetic factors that play with environmental factors, so it's a combination of multifactorial disease that leads to the development of cancer. Now, why start to look at genetics, or why study families? Well, if we can identify genetic variants, maybe we can predict the risk of Barrett's better and identify those patients who cannot be identified by symptoms alone. If we can identify the major genes, then we'll be able to explain the biology of this disease and give more targeted insights and perhaps find molecules that we can target with chemoprevention instead of using aspirin and PPIs, maybe even get better therapeutic targets, and then be able to define biomarkers for progression to Barrett's and cancers. Our interests actually started about 2 decades ago when we started this case control study that showed that indeed Barrett's and esophageal cancer did aggregate in families, And we started looking at a syndrome that we defined as familial Barrett's esophagus, where Barrett's esophageal cancer and junctional cancer were present in 2 or more family members. We found in a large prospective study with a lot of friends around the country that Barrett's tend to be familial in about 6% of patients. Cancers tended to be familial in 10%, so maybe there's something about malignancy that has a greater genetic susceptibility. And then one of the ways you tell whether something's genetic or not is you look at age of onset. And the age of onset of cancers in families where there were 3 or more affecteds tended to be about 5 years younger, and perhaps we should be more considerate of families that have 3 or more members. Their screening maybe should be done at an early age, and that's also the group that we need to concentrate on if we're going to make some genetic advances in this disease. So what kind of approaches can be used to identify susceptibility genes for diseases? Well, one approach, the traditional older approach, Mendel counting the yellow and green peas, is to do a linkage study and analyze multiple pedigrees. That is the strategy that's been used for identifying APC in familial colon cancer. It was a strategy used for identifying BRCA1 and BRCA2 in breast cancer. Unfortunately, esophageal cancer, perhaps because it's an inflammatory cancer, it turns out to be much more heterogeneous. We have identified linkage, but there's no major gene out there to be found in those linkage areas, so we're not going to have a BRCA1 or BRCA2 that comes out of it. GWAS studies are great. There's been a great enthusiasm for them. They are waning a little bit. This is an approach where you do a case control studies of thousands of subjects, you genotype millions of SNPs, and you identify associations with SNPs. These often identify SNPs that are common and give you a lot of attributable disease in the population, but the effects of these SNPs, the odds ratios tend to be about 1.5 or less, so they don't give you major effects. They are useful, and they can be used for developing better prediction models. And then the big advance in this disease has been next-gen sequencing, which is what's given rise to personalized medicine. And next-gen sequencing you can do in one large exceptional family, and that was a strategy used by Bert Vogelstein for identifying PALB2 in familial pancreatic cancer, and we'll talk about how we've used that strategy. So there have been a number of meta-analyses done now in Europe, Australia, and the United States, and then meta-analysis combining all those data. We now have nearly 20 SNPs. Notice the odds ratio are about 0.8 to 1.5. Many of them are near genes with regions of interest, but how these SNPs actually lead to disease, the functional value has still not been sorted out. We did one study of this large exceptional family that took a number of years to find out, and when next-gen sequencing came around, we identified the first gene named V-SIG10L in this family from sequencing 4 members. The blue members of those family had Barrett's esophagus. The ones with red with a cross through them are deceased members who had esophageal cancer, so you can see a family that has a striking dominant penetration. And this gene, V-SIG10L, nothing's known about this disease. It's one of the 10 most highly expressed genes in the normal esophagus. It's a gene that's only expressed in the cervix and a little bit of the skin, so it's a gene that is primarily expressed in squamous tissue in the esophagus. When you put this gene into 3D cultures, the normal gene, you get a normal 3D culture with a stratified squamous epithelium. But when you put the mutated gene that we had found from that family, it disrupted that normal 3D architecture in the 3D culture. We also were able to get some squamous tissue from the proximal esophagus of a family member who carried this mutation, and he was on a PPI, and you can see on the lower right-hand corner that the intercellular spaces in the normal esophagus and the proximal esophagus showed large dilated intercellular space, and a complete absence of desmosomes. So we don't know exactly what this gene does, but it seems to affect the normal squamous epithelium and make it susceptible to progressing to Barrett's. Another neat approach that takes advantage of the racial disparity was one that David Beer did. We collaborate with David Beer as a part of a consortium called BetterNet. And what David reasons is Barrett's and esophageal cancer are rare in African Americans, and perhaps this again gives us a clue to the genetic susceptibility to esophageal damage. And he just got some normal esophageal biopsy samples from Caucasians and African American patients and genotyped them using mRNA expression arrays. And lo and behold, he found this gene, glutathione ST2, which was 5-fold higher expressed in African American squamous esophagus than Caucasian squamous esophagus. This gene is protective against DNA damage and may protect African Americans from damage with reflux, and we're looking forward to seeing what it does. So to summarize our genetic studies, and I'm 30 seconds over, GWASs have made some progress and have identified SNPs with odds ratios of 0.8 to 1.2. The functional studies of those SNPs are still pending. There are some preliminary prediction models that incorporate those SNPs. Those models need to be improved to where we can use these SNPs clinically in predicting who's going to get Barrett's esophagus. From our family studies that have ongoing, we've discovered one gene that seems to be important in maintaining normal squamous esophagus. And then finally, using the advantage of racial disparity, we've identified a second gene which seems to protect the esophageal cells from reflux-induced oxidative damage. I'll thank you for listening.